Okay, this is the main video for Chapter 7. It's going to cover mutations and recombination, and it's going to take us into the biotechnology Chapter 8. So first let me start out with the difference between a mutation and recombination. Those are the two things in this chapter we need to talk about. A mutation is a change-up in base sequence of the DNA, of the genome. It can be in RNA, if RNA is the genome, but there's only one thing that can have an RNA genome. There are some viruses that do. We'll talk about mutations in viruses when we get into viruses. But for right now, just remember that it's in DNA. So, if there's a change, well, let me back up. So, let me compare that with recombination. Recombination is new DNA being added to existing DNA in an organism, recombining at least two different genomes together. Whereas, again, a mutation is a change in the existing genome. There's no new DNA added. Alright, so when there is a mutation, a few things can happen. A, nothing may happen. There may be no change in the protein coded for by the DNA, by the gene. Or, if there is a change in the protein, it may be a minor change, but it still functions. Or it can be a major damage to the protein, and it doesn't work at all. So, on the picture that you see here, you're looking at a section of DNA. Whoops, let me get into the correct mode here. Get back. Alright. Alright, so here is the sequence of DNA. There's a little three-letter word there, TAC and ATG on the other side. And remember that during, if you look right over here, here is the normal DNA replication. And again, here would be the sequence that you're looking at. And this is what should be produced during the replication. Okay, remember that these two strands separate out right here, and then each one, the two strands of the DNA separate. Each one makes a new strand. And they'll be the same. They'll both be TAC and ATC on this side. Okay, now, the sense strand of the DNA is this bottom strand, and it then is involved in the production of mRNA, which you see right here. And if your codon, if your three-word base sequence in DNA is ATG, then your codon in mRNA will be UAC. Let's say it codes for the amino acid tyrosine. And that tyrosine is part of whatever protein is being made here at the bottom. Alright, wild type. That's your normal run-of-the-mill protein. Now, look over here to the left and see what has happened. Now, let me erase this. And let's say that this is the strand that is replicating during the DNA. That strand is that strand right there. And during this production, there is a change in the base. Look at the A right there. And of course, that changes the other strand. And we now have a different section of a double strand than we had up here at the top. That codes for a codon, UAU, which may also, luckily, code for tyrosine, just as UAC does. Because remember, there's redundancy in the code. There may be 3, 2, 6 even. I think for proline, there are about 6 different, or isoleucine, there are about 6 different codons. They all code for the same amino acid. So you could luck out and end up with the same amino acid, and you have not changed the protein at all. That's called a silent mutation. Now, go over to the left one more, and you see that here, as a result of that change in the DNA, trying to figure out why am I right. There it is. As a result of the change in the DNA, we now get a codon UAG, and UAG, remember, is a stop codon. So when the ribosome gets to UAG, whether UAG should be there or not, improperly, it stops. It thinks it's at the end of the mRNA, finished coding for a protein. So now you get an incomplete protein, and that's called a nonsense because obviously that protein is not going to be functional. Then, lastly, you go over here on the far left, and we end up with a change in the base sequence such that we now get a codon that codes for a different amino acid, and maybe it does not totally destroy the protein function, or it may change it in some way, or it could damage it so it doesn't work at all. That's called a missense mutation because we've changed the amino acid on it. So you can see that you can have anything from no effect to what is sometimes called a sublethal mutation. Doesn't really do much damage to it. Cell still lives and can work. Or it can be a lethal mutation, and whatever the protein is being made that's defective, the cell can't live with it and it dies. Alright, now everything you saw right over here was a point mutation. So all we did is we changed one base. The other two bases in the codon, for example, of the mRNA, have not been affected. Okay, point mutations. Look, they're the most common. Alright, so there are substitutions. I just took you through that, where we substitute one base for another. Alright, now the other one, a little bit more devastating, is a frameshift mutation. And this would be exemplified by an insertion or a deletion. And let me take you to the next page. And look over to, most of this is stuff we've already talked about, but look over here. Alright, so you see the sequence of double-stranded DNA, and notice that, okay, here's the normal up here, and look right there between those two adenines. And notice right here there is an insertion of thymine with its base pair on the other side. We now have an extra codon change. The codon now, note the first codon was UUU here. It's still UUU here, so it hasn't changed. The next codon would have been UAU, coding for tyrosine, but instead, because of that base pair added, we now have a codon AUA, which doesn't code for tyrosine, it codes for isoleucine. The next codon would have been GCA, but it's not. It's now UGU, which codes for cysteine. So in other words, every single codon down the line now is going to be changed. We have shifted the frame of the codons by one base to the right. Now, on the bottom, right below that, you see a deletion. And here, base pair is taken out during the DNA replication. So now, all of the codons are shifted to the left, and all the amino acids from that point onward are wrong. So this is usually more devastating to the cell, causing death. Alright, so something that causes a mutation is called a mutagen. Alright, so let me back up. Alright, so let me talk about spontaneous versus mutagenic induced mutations, that is. Cells screw up in their replication when they're putting new bases, growing a new DNA strand. They make mistakes. In bacteria, it's estimated that a bacterial cell, when it's replicating its DNA, about 1 out of every 100 million bases during the replication is wrong. In eukaryotic cells, like human cells, they estimate that it's about 1 out of every 100,000 to 1 out of 1 million is wrong. Those are spontaneous mutations. There's nothing that causes them, nothing that has induced them, just a screw-up of the cell. Now, we don't so much care about spontaneous mutations. They're rather rare. We're more interested in ones that are caused, and those are called induced mutations, and they're caused by mutagens or mutagenic agents. So, you have chemical mutagens, you've got radiation that is mutagenic. In the realm of radiation, you've got ionizing radiation like X-ray and gamma. And in UV radiation, of course, we're talking about ultraviolet. Ultraviolet radiation causes adjacent thymines to bond to each other. Let me draw a section because I don't have an example here. So here is a DNA double strand, and let's say, let me just put a base sequence here, and let me put the strand on the other side. It's sequence. Okay, so remember that there should be hydrogen bonding right here between, I can't quite get it to do it, hydrogen bonding right here between the bases holding the two strands together. But what happens in UV radiation being a mutagen is when you have two adjacent thymines, like right here, a bond forms between them. Change into a red color here. And they form a bond. It's called a thymine dimer, and that messes up the attachment to the other strand, and then it messes up the replication. That's UV radiation's effect. Ionizing radiation does a lot more damage. It doesn't do it by thymine dimers. It does it by affecting the entire sequence of the DNA. Now, chemical mutagens. You've got lots of different types of chemicals that are mutagenic. Red dye number 2, acridine orange, nitrous oxide, even some things like caffeine, such like that, nicotine. We're not going to get into all the details about how these mutagenic agents work. I mentioned before that repairs can be performed. If they're spontaneous generations, they're not occurring that often, and so when there's a mess up, there's a enzyme that comes along and fixes it if it's been identified and it hasn't occurred too frequently. But when you've got induced mutations and they're occurring at a much faster rate, not all those mutations can be identified, and when that mutation stays and it's not repaired, then as that cell replicates, if it can still live with that mutation, the daughter cells will also get that mutation. It's now become part of the genome. Okay, so there's a test using bacteria for determining mutagenicity. In other words, this is a test that determines if a chemical is mutagenic, but instead of using human cells or animal cells or whole animals, we're going to use bacteria. The idea is that if a chemical is mutagenic for a bacterial cell, it is likely that it's also mutagenic for an animal cell, because frankly, DNA replication and protein synthesis are pretty common between prokaryotic and eukaryotic. Okay, now, you know what a wild-type organism is. It's your, run-of-the-mill, normal guy that's out there living. Okay, but for this test, we're going to need a mutant. We're going to use what is called a histidine mutant. A histidine mutant, I'm going to write it out. Histidine is one of the amino acids, and usually it is written as his negative. In other words, it is lacking the gene to be able to make its own histidine. Bacteria can usually make their own amino acids. Histidine is one of the ones they can make, therefore they make their proteins. But if you're a his negative E. coli or Salmonella, and you are lacking the gene to make histidine, and you can't make your proteins properly because you need all 20 of the amino acids to make the proteins, then that means you can't make the protein, and odds are you can't live and you're going to die. So we're going to use a mutant missing the gene for histidine production. It's called an auxotroph. An auxotroph is a metabolically deficient mutant. Because you can have lots of different mutants. You can have mutants that make more alcohol. You can have mutants that make antibiotics. Okay, so we are going to use for this test an auxotroph that is lacking the ability to make histidine. If it's not provided histidine in the media, it can't grow. If histidine is put in the media, it will grow. We're going to use a salmonella auxotroph. Let me show you what that would look like, at least write it out anyway. It will look like this. It's written out as a salmonella. That's its genus name. His negative. It's lacking the gene to make histidine. We use this particular mutant because, well, you'll see why. This is a reverse mutation, and this particular mutant, its his negative mutation can easily be reversed. In fact, the way we made the salmonella was a mutation to begin with, but I'm not going to get into all that. Okay, so I'm leading you to the Ames test, which is the test using bacteria for determination of mutagenicity. You see the test in the picture up here. So what we're going to do is we are going to streak our salmonella onto the agar plate. Okay, so we take a loop or a swab, and we swab it all the way across the plate. All right, in red. Now, we take our chemical that we want to determine is mutagenic, and we saturate a little cotton disc, and we put it in the middle of the plate. So the whole plate's covered with salmonella. It's not grown yet. We just inoculated it. We put the chemical right there. We now incubate. All right, so remember I mentioned, by the way, this media does not have histidine in it. Remember, I mentioned that this particular mutant, auxotroph of salmonella, is missing the ability to make histidine. It will not grow. It can't make its own. You have to provide it in the media. If you don't provide it in the media, it won't grow. Okay, so this medium does not have it. So, when you inoculate it and you incubate it, you might get some spontaneous mutants here. Remember that bacteria produce mutations spontaneously about once every 10 to the 8th base additions. But there's really not any significant amount of the growth of bacteria. But look down here at the bottom, and you'll notice bunches and bunches of colonies growing right around the chemical. The chemical is having an effect. It is reversing the mutation. That's why this is called a mutational reversion assay. Assay means test. This is a weak mutant that will revert back to wild type if the chemical is mutagenic. Now, why do we care about mutagenicity? There is a high correlation between mutagenicity and carcinogenicity. So if an agent is mutagenic, it is likely it is carcinogenic, cancer-causing. If it's mutagenic and carcinogenic, well, let me back up. If it's mutagenic in bacteria, it's probably going to be mutagenic in eukaryotic cells like humans, which means, again, because of that high correlation, it's going to be carcinogenic in human cells. So that's the Ames test. A lot easier than using animals and cells and such. Okay, so let's move to recombination. Here's the overall view. All right, here is, over on our right, let's say here is a staph. Well, it probably wouldn't be a staph because staph's not a bacillus, but let's go with it anyway. And chromosomes in blue. And over here on the left is an E. coli, and its chromosome is in pink. And what we're going to do is we are now going to destroy the E. coli cell, and we're going to take this DNA, and we're going to cut it into pieces, and we're going to add a section to this cell, and that's what you see right here. So a piece of E. coli genome has been added to the staph. Now, one of two things can happen. Look at the left. Bacteria have defensive mechanisms to destroy DNA that's not supposed to be in the cell. So the DNA that was put into the cell could be destroyed. No effect. Okay? So we're back to the original bacterium. No change. Scenario on the right, which is what we're really interested in, is that the DNA inserts into the chromosome, is not destroyed, and it now becomes part of the host cell genome. Because we're going to be moving into biotechnology, which is the manipulation of cell genomes. And obviously we don't want that to happen because that would cost us a lot of money and time. We want this to happen. We want the recipient cell to pick up the DNA. Okay, so that's kind of the big picture of where we're headed. All right, so some things you need to know. I've already taken you through the difference between the two terms. When we put in new DNA into the recipient cell, it is called a recombinant for obvious reasons. Now, vertical versus horizontal gene transfer. Let me go back to this cell. Look at this cell on the right-hand side at the bottom. Let's say this is a staph cell. It's now got new genetic info, as you can see. When it divides into daughter cells, the daughter cells will look just like it. The daughter cells will have that red chromosomal material. Their daughter cells will. So in other words, the new DNA in red is now part of the genome of all the daughter cells. All right, that is called vertical transfer to the daughter cells. Now what we're more interested in is horizontal gene transfer, where humans, where we can, in a lab, put in new information into the host cell, like we could put a plasmid in, or maybe we could just take some double-stranded DNA and put it into the cell. All right? Now, we can do this lots of different ways. Here are the three ways: transformation, transduction, conjugation. So this cell receives DNA by horizontal gene transfer. Now, if the DNA stays in the cell, when this cell replicates and it produces daughter cells by binary fission, its daughter cells will have the same DNA. But these three mechanisms that I'm going to go through are part of horizontal gene transfer. Okay, so we're not going to spend very much time on this. All right, here are the three ways we're going to do it. Let me just go ahead and go with these. All right, so the first one is transformation. I'm going to go into what you see on the diagram here in class. I'm not going to go into it now, although it's covered in your book. In transformation, we have a cell. Here's our recipient cell. Okay, here's its normal DNA right there. All right. And let me change colors into blue. All right, so what we're going to do is we're either going to take a piece of DNA, or I can take a whole plasmid and I can put it into this recipient cell. Okay, so here's a little plasmid right here. Or, again, it could be double-stranded DNA, just a little fragment of it. So, what do we have to do? We have to do something to the cell wall. We don't destroy the cell wall, but we make the cell wall a little bit more porous so that the DNA can go through it. And remember, there's a cell membrane inside of here. So the cell membrane has to become more permeable to the DNA. The DNA gets into the cell. There we are. There's horizontal transfer. This is going to come up in the biotech chapter. Why is it important? Well, it's going to become important to us because we use this mechanism a lot in recombinant DNA technology. But realize that this happens a lot in nature. There's a lot of DNA lying around the place from cells that are dead. The DNA can last hundreds and hundreds and hundreds and hundreds of years or longer. and the DNA can get into the cells. So what does this do for the cell? It increases its chances of survival. Anytime there's a change in DNA, new DNA added, you get genetic variation and potential survivability advantages. So remember, although we're headed towards manipulation in a lab for DNA technology, this happens in nature also. Okay, so let's go to the second mechanism. Second mechanism involves a virus. Transduction. The name for viruses, phage. Phage means virus. Okay, now look over here, and let me take you through what a virus does. There's a virus. By the way, this is a bacterial virus. That's a bacterial cell right there. Look inside of the head of the virus. Its DNA is in green. A virus injects its DNA into the host cell. There you see it coming into the new host cell, the bacterium. Bacterial chromosomes in purple right there. During the takeover by the virus, the bacterial chromosome is broken down into pieces. You see right there. Then new viruses are produced inside the host cell. You can see about 6 little baby viruses that have been produced over here. But look at this guy right here. This guy is different. He has the wrong DNA. In his head is packaged a piece of this bacterial cell's chromosome. The rest of these viruses got viral nucleic acid. That virus got the wrong nucleic acid. Now, this cell is a goner, okay? He's dead because the viruses are now going to burst out. Each of these other viruses goes out to infect new cells. But think about this guy right there. He now tries to infect a new bacterial cell, but he doesn't have the right viral genetic information to do so. Remember that he's carrying packaged DNA from the previous bacterial cell. That genetic information goes into the host cell. You see it in purple. It can become part of the host cell chromosome. We now have a recombinant bacterial cell. It has its own genetic info. It's got the genetic info that came from the original bacterial cell over here. We're not going to get into the two kinds, generalized versus specialized. Again, this is going to come up in the biotech chapter. It's used in recombinant DNA technology to engineer cells, but it also happens in nature because there are a lot of bacteriophages, viruses that infect bacteria. So this is not an odd thing to happen. Okay, third scenario, bacterial conjugation. All right, so look at this scenario right here. We've got a bacterial cell, cell 1. His host cell chromosome in blue, plasmid in pink. During this process that you see called sexual reproduction, also called conjugation, same conjugation, same thing, two different ways to say it, a pilus attaches to a new cell. Now, this pilus came from the cell on the left. It had a fertility gene on the plasmid. That fertility gene codes for the production of a pilus. Now, these two guys are hooked up. Notice right here that the plasmid is replicating, and that new replicant DNA of the plasmid now has ended up in the recipient. You see it right over here on the far right. Now, notice the headings over here. The guy that has the plasmid is called an F+. The guy that doesn't have a plasmid yet is F-. F is for fertility. When this plasmid now goes to this F-, he's become an F+. By the way, plasmids can not only be gained, as you see here, plasmids can also be lost. Now, there are other variations on a theme here. You can have, down here in the second scenario, this plasmid has a piece of gene from the host cell chromosome, and when it ends up over here, in this recipient cell, there has been a transfer of this gene right here that came from this cell over to that cell. Whereas up here on the top row, only genes from the plasmid ended up in the recipient. And then at the bottom, there's another a different variation where the whole plasmid inserts into the host cell chromosome. Okay, why important? Again, this can be used in a laboratory for manipulation of bacteria. It also happens in nature. Okay, that's the end of Chapter 7.